My name is Parth Patel. I am a postdoc at Wasson Lab, University of Saskatchewan, Canada. The topic of presentation is microfluidics approach for the one-step preparation of subunit vaccine for bovine viral diarrhea virus. The bovine viral diarrhea is a major indigenous viral disease in cattle caused by bovine viral diarrhea virus. BVDV is a member of genus Pestivirus, family Flaviviridae. It causes bovine respiratory diseases complex and reproductive dysfunctions, which define BVDV as one of the most economically significant pathogens to the cattle industry. Inactivated and modified live vaccines were prepared against BVDV infection, but both vaccines have significant shortcomings. The potential and safer alternative is to prepare effective subunit vaccine. The antigen used here to prepare subunit vaccine is referred as E2 protein, which is made up of 371 amino acids. The open reading frame encoding E2 protein was modified to express a protein with N-terminal signal peptide of tissue plasminogen activator to enhance the induction of both antibody and cellular immune responses in animal models. An histidine tag was added to C-terminal of protein to purify it. Adjuvants are crucial components of vaccine and play an important role by improving immunogenicity of vaccines. In current research work, recently developed tri-adjuvant platform, a combination of polyphosphazine, referred as PCP, innate defense regulator peptide, referred as IDR peptide, and poly-IC, have been employed to prepare the vaccine. PCP is a synthetic water-soluble polymer having an immunomodulatory property. Poly-IC is a toll-like receptor which are highly expressed on myeloid dendritic cells and facilitates cross-presentation of antigens. The IDR peptide has microcidal and an immunomodulatory property. It is widely accepted that an intranasal administration of vaccine induces systemic and local immunity, which provides an increased level of protection against respiratory disease. In our prior research, cationic lipid formulation of tri-adjuvant has successfully demonstrated its ability to activate immune response following nasal administration. To prepare subunit vaccine particle containing E2 protein and tri-adjuvant, a combination of DOTAP, a cationic unsaturated lipid, and DOPE, a zwitterionic helper lipid, were used in 1:1 molar ratio using microfluidics methods, which are now being increasingly employed for its controlled microenvironment to prepare lipidic nanoparticles in an easy, cost-effective, and reproducible manner. To prepare the vaccine formulation, a 3-channel microfluidic droplet junction chip was used to achieve microfluidics mixing, where each channel was connected to its own computer control pump. The IDR peptide mixed with E2 protein was kept in pump 1, poly-IC mixed with PCEP was kept in pump 2, lipids DOTAP and DOP were dissolved in ethanol and kept in pump 3. The rationale behind such arrangement of components is to avoid blockage of the instrument tubings. At the initial stage of optimization using microfluidics platform, it was observed that if IDR peptide and poly-IC are kept in the same vial, they do likely to form precipitates, which were responsible for the blockage of the tubing. The flow for pumps 1, 2, and 3 were set to 33, 11, and 13.9 microliters per minute respectively. The flow rate ratio was 4, and total flow rate was 58 microliters per minute. The temperature control unit was employed to maintain chip temperature at 1 degree Celsius to facilitate vaccine particle fabrication, as the transition temperature of DOTAP lipid is less than 5 degrees Celsius. The concentration of components were optimized as the final 20 microliter of vaccine formulation should contain 1 microgram of E2 protein, 5 micrograms of poly-IC, 10 micrograms of IDR peptide, 5 micrograms of PCEP, and 256 micrograms of total lipid. The weight ratio of poly-IC, IDR peptide, and PCEP was set to 1:2:2:1, which was established by extensive in vivo studies. The lipids to tri-adjuvant ratio was decided based on phosphorus ratio. When tri-adjuvant and E2 protein encounters at T junction, PCEP immediately covers the rest 3 components to form antigen tri-adjuvant as an anionic intermediate product, which will then encounter mixture of cationic and helper lipid at the junction on chip. At the junction on chip, lipids are likely to precipitate caused by ethanol diffusion and cover the whole antigen tri-adjuvant complex to form lipidic nanoparticles. Having lipids on the surface an antigen triadjuvant in the core of the particle, which is referred as LAT subunit vaccine formulation. The freshly prepared LAT formulation is subjected to nitrogen flow at 40 degrees Celsius for 15 minutes to remove ethanol. Another form of subunit vaccine, referred as LT plus A, was prepared by batch method. Here the batch method refers to the preparation of formulations by stepwise addition of components. To prepare LT plus A subunit vaccine, IDR peptide was mixed with poly-IC and then PCP was added, which was followed by addition of preformed liposome and then antigen was added at last. The components were added in required weight ratio to maintain respective concentration in 20 microliters dose of the vaccine. The major difference between the two forms of vaccine particle is the arrangement of an antigen. In LAT form of vaccine, antigen is entrapped in the core of the vaccine particle. In contrast, LT plus A form of vaccine, where antigen is attached on the surface of the vaccine particle. The E2 protein in PBS and triadjuvant and antigen triadjuvant prepared by batch method have shown polydispersity index greater than 0.5, irrespective of their size, reveal their non-homogeneous nature. Liposomes were found cationic and homogeneous in nature with mean diameter of approximately 100 nanometers. The LT plus A vaccine particle was found having significant laser positive zeta potential than liposomes, as LT plus A vaccine has antigen triadjuvant attached on the surface of the liposome. An anionic zeta potential of antigen triadjuvant neutralized some of the cationic zeta potential of the liposomes. In contrast, LAT vaccine particle have significant higher cationic zeta potential than LT plus A vaccine particle, because in case of LAT vaccine, the antigen triadjuvant complex is entrapped in the core of vaccine particle and do not contribute to neutralize the cationic zeta potential of the lipids located on surface of the particle. The LT plus A vaccine particles form gross visible aggregation and sedimentation occurs, which was also observed in transmission electron microscopy studies. In contrast to that, the LAT vaccine particles remain suspended and DLS results prove its uniform and homogeneous nature. In 10 studies, antigens are observed as black dots inside the artifacts and antigen aggregates can be seen in the image. The 10 images of lipid triadjuvant and LT plus A vaccine particles prepared by batch methods were compared to define the role of antigen in the formation of visible gross aggregation of the particles. Lipid triadjuvant is shown on the right side, where lipid layers of the liposome and triadjuvant attached on the surface of the liposome are seen clearly. No aggregates of the lipid triadjuvant particle was observed under TAM, and also no visible gross aggregation occurs. When antigens are added to the lipid triadjuvant formulation to prepare LT plus A vaccine particle, the antigens have started attaching on the surface of lipid triadjuvant particles and eventually form aggregates and sedimented. In the TAM image, on the upper left side, we can see individual LT plus A vaccine particle where antigen triadjuvant complex is attached on the surface of the liposome, and the aggregates of the LT plus A vaccine particle can be seen on the lower left side of the slide, where lipid layers are clearly visible on the outer side of the aggregate. Lipid triadjuvant was also prepared using microfluidics platform to compare with LAT subunit vaccine particle. In both the cases, triadjuvant and antigen triadjuvant were entrapped in the core of the lipid triadjuvant and LAT vaccine particles respectively. Although the data is not shown here, size difference was observed between two formulations, where LAT vaccine particle has significant higher mean diameter than lipid triadjuvant particles. The lipids arranged in layers on the surface of both the particles were observed clearly as shown in the image. The surface plasmon resonant experiments to demonstrate LAT vaccine particles binding efficiency to type 2 mucin from porcine stomach are being conducted. The comparative in vivo studies have completed and ELISA analysis of the collected blood samples are in progress. To sum up, based on the characterization data of LAT vaccine particles, it was concluded that microfluidics is an advanced approach to prepare monodispersed lipidic nanoparticle in a single step and in controlled manner. The cationic lipid incorporated in the preparation of subunit vaccines have played a crucial role to engineer stable cationic lipid nanoparticles. The effect of two different arrangements of antigen triadjuvant was observed on physical characterization data of the subunit vaccine particles, and its effect on immune system will be revealed based on in vivo study results.